Thank you everyone for being here. I hope you enjoyed the great talks we had this morning, and more importantly, I hope you enjoyed the food. I'll be telling you a bit about our experience at ProImmune with regards to looking at wanted immune responses and characterizing epitopes, not only to develop targeted therapies, but also as an essential tool to monitor those responses to your treatments. So I'll try to figure out how this works, actually. There. All right, so just a quick background. In the context of cancer, as you all know, the purpose of cancer immunotherapy is to actually harness an anti-tumor specific immune response, usually in a context where the immune response is actually dampened. Now, if you want to trigger this immune response, a lot of therapies actually are targeting certain tumor-associated antigens. Now, to be able to target those TAAs, as they call them, you actually need to know what they are. And therefore, the identification and the characterization of those tumor-associated antigens is crucial for not only the development of your targeted therapies, but also further down the line to investigate the immune response that is triggered by your treatment, and characterize all this immune response as a whole with immune monitoring tools. Now, at ProImmune, we have this experience with actually a broad experience, and we've been around for a few years now, looking at specific immune responses, whether it's through Tetramer tools or Pentamer tools, so all the multimer kind of category, but also we've developed some crucial mass-spec-based technology to identify a lot of those epitopes, and I'll be detailing some of those technologies. With regards to cancer therapy in particular as well, we have worked over the years on a broad range of cancer therapies, and therefore have become experts actually in the identification of those responses to your drug, or to the tumor epitopes that have been identified. Now, just a quick reminder, and I won't be telling anybody anything new here, but the key to triggering an immune response is, at least on the adaptive level, is by having your T cells recognize here a peptide that is presented at the surface of the cells by an MHC molecule here in blue. So this recognition by the T cell receptor of this MHC plus peptide complex is absolutely necessary to trigger a subsequent immune response. And this is basically what we specialize at in ProImmune, and it's the study of this binding between the peptide and the MHC peptide complex. Now, ProPresent is, I won't detail this too much because Jeremy is going to talk about it further tomorrow, but very briefly, ProPresent is a mass spec based assay that allows you to identify what your cells are presenting at their surface. And this is typically, you can't get any better at epitope identification. What we do is that we take the cells, immunoprecipitate this MHC plus peptide complex, and then isolate the peptide from the MHC groove, and then identify the peptide sequences by mass spectrometry, and we will report the peptide sequences as they are presented. We can use a variety of cell sources, whether it's the actual tumor tissue in the context of cancer. We can also use tumor cell lines, healthy donor cells. We're actually very flexible with the cell format that you can send us. And as you know, CD8 responses are crucial in the case of tumor immunity, but increasing evidence is showing that CD4 responses are also important and shouldn't be neglected, and therefore we can look at class 1 presentation that would trigger a CD8 response, but also class 2 presentation, which will trigger a CD4 response. We have another format of the assay that looks at the antigen presentation from therapeutics that you can develop, and that's something that Jeremy will be developing tomorrow, detailing tomorrow. The greatness of this technology as well is not only that it's mass spec based and we can identify the peptides by their sequence, but we can also look at modifications of those peptides if you know what you're looking for. We can look at phosphorylation, we can look at other chemical modifications and post-translational modifications. So this is one of the great technology that we have developed that will allow you to identify potential targets for your therapeutics, for your vaccine development, or even for your further on immune monitoring assessment of a therapeutic. Just to illustrate how ProPresent has managed to help a study using parvovirus for the treatment of glioblastoma. I'll give you a bit of background here about the actual study that was performed by DKFZ in collaboration with Oryx. And the study was published last year and actually in Molecular Therapy, which as an oncolytic virotherapist myself is a great journal. So I definitely recommend for you to read the paper. So they came to us with this therapy that they had developed. They had treated glioblastoma patients with an oncolytic parvovirus, and they have different regimes with IT, so intratumoral and intracranial injections of the virus, and managed to harvest the PBMCs of their patients. Now they needed to identify the response to their treatment and wanted to look at how the patient had responded to the virus, but also how the patient has responded to the tumor in the context of the therapy. Now, the easiest way you can think of is to just take the cells from the patient, immunoprecipitate the peptide plus MHC complex, and look at the epitopes that were presented at the surface of those patient cells. The issue was that they didn't have enough cells to give us so that we can do the analysis for the epitope, the peptidome that was presented at the surface of those patient cells, and they didn't have enough to do the immune monitoring either, so we had to choose something. So we sat with them, had a chat, looked at what they had available, and it turns out that they had a broad library of cancer cells, glioblastoma cancer cell lines. So what we suggested was just for them to infect those cancer cell lines with the parvovirus and send them to us. Now, it was a bit of a long shot for the simple reason that we needed those cancer cell lines to match the HLA type of the patients, because otherwise, given the HLA restriction of certain epitopes, it wouldn't be a good match if the cell lines didn't match the patient's HLA type. Nonetheless, we were successful. We managed to find a good selection of cancer cell lines. They sent us the parvovirus-infected glioblastoma cell lines, and we used ProPresent to just immunoprecipitate the MHC plus peptide complex and identify 5 peptides from 2 proteins of the parvovirus and 20 peptides from 18 proteins of the glioblastoma, or at least proteins that were associated with glioblastoma. So further down the line, I mean, identifying those epitopes is crucial for the development of the therapies, but also looking at what sort of response we're getting to those epitopes. But it's also important to qualify the functionality of those epitopes, and therefore immune monitoring is crucial here to confirm the immunogenicity of those epitopes and whether they're functional epitopes. So we performed an ELISpot assay for them. And just a quick reminder of what an ELISpot assay is. We culture cells with the given peptides or antigens that you're interested in looking at. And then we quantify the interferon gamma response and look at basically CD8 and CD4 T cell responses using this assay. And in this case, they have managed to confirm basically that the peptides that we had identified with ProPresent from the glioblastoma were actually functional and had triggered an anti-tumor immune response. In parallel, the parvoviral proteins that we had identified as well with ProPresent had also generated a positive response. As you can see here in the blue bar is the parvoviral, on the top, it's the parvoviral epitopes, and in black and gray, those are the glioma. So not only did we manage to identify functional epitopes that were by ProPresent, but they also confirmed that there was an anti-tumor response that was triggered by their parvoviral therapy. So that's really one of the, a nice case study to kind of show how ProPresent has managed to help them identify the response to a therapy. ELISpot assays have managed to confirm the response. There are other ways for immune monitoring that we can use, and actually ProImmune is specialized in the development of Pentamers, which is class 1 multimers, that will allow you to look at CD8 epitope-specific responses in the context of a therapy or in the context of, well, here if we're talking about cancer. So we're very proud of this technology. It's been around for over 15 years now, and we're the first commercial providers of this technology. The structure of the multimers itself is actually quite simple. It consists of 5 MHC plus peptide complexes, which artificially basically provide this necessary link with the TCR. The 5 MHC plus peptide complexes are linked by a coiled coil domain to 5 fluorescent tags, which allow you to monitor and quantify those T cell populations by flow cytometry. So far, it's the most consistent and robust technology, and we have been, we're very proud to say that we have thousands of publications mentioning this technology. What's great about it as well, it's the flexibility of staining. You can stain them, you can have them stained with biotin, RP or APC, and we can deliver them in a variety of test sizes. And we've been around for such a long time that we've managed to develop over 450 catalog items, 200 of which are actually cancer specific. So because they're because they're catalog items, they can be delivered to you in a very, very quick time. 1 or 2 days for the most popular items, but 1 or 2 weeks for the less popular. If amongst this long list of items, your preferred combination of MHC plus peptide complex is not available, we can also custom make them for you. To illustrate how Pentamers can help in the immune monitoring of responses, I'd like to tell you about this Dana-Farber study that happened using adoptive transfer of T cells. And so in this case, the team had developed an adoptive transfer of patients' CD8 T cells that are specific to a tumor antigen. They would harvest them from malignant melanoma patients, educate them to recognize the tumor-associated antigen, and reinfuse them. Now, while the therapy had a great therapeutic potential, unfortunately, the cells were short-lived once reinfused. So they needed to improve the longevity of those infused cells. And what they did was they managed to develop a protocol that generated effector memory T cells using artificial APCs, so antigen-presenting cells, where they basically educated the cells to recognize a given tumor-associated antigen. In this case, it was MART, and generate an effector memory pool. And they then used to look at the efficiency of their treatment and their protocol. They used our Pentamer, class 1 Pentamers, to monitor and quantify the populations at different time points. they realized that after they infused twice at 35-day interval, they re-infused them at 35-day interval. The cells were quite at day 0 and day 35. They were still present, but they managed to still detect with the pentamer some of the March-specific populations at day 126. And their protocol was actually even further improved when the adoptive transfer was used in combination with a monoclonal antibody for anti-CTLA-4, where, as you can see, the combination with anti-CTLA-4 has managed to prolong the viability of the cells and their presence in the circulation up to 537 days. So that's really an example of how Pentamers can allow you to track basically the response of your to your treatment, and in this case quantify your populations. This is just another example of staining. We like this dot plot because it corresponds to a case study that I will mention towards the end of my talk. And in this case, it's an epitope that's specific to an HIV epitope. So similarly to class 1 and the study of CD8 responses, it's also important to look at CD4s, whether it's in the context of cancer or in the context of infectious diseases. And I'm pretty sure a lot of you have also investigated the responses to CD4, but I do want to stress the importance of CD4s in the context of cancer too. And for that, we also provide, similarly, tetramers. So it's what we call the ProT2 MHC class 2 tetramers, which are MHC class 2 molecules that bear the epitope of choice. In this case, we have as well catalog items that cover a broad range of specificities, going from flu to EBV. But we also have a broad range of cancer epitopes available as our catalog items. Again, here we can custom synthesize the items if you don't have your chosen epitope amongst our list, and again on a broad range of alleles, of human HLAs basically. We also provide biotinylated monomers for those who are more interested in stimulation and things like that. I won't detail that here, but if you're interested, please come to see me. An example of staining for the class 2 monomers, and this you'll recognize Professor Jörg Laurer's name from this morning. This is just a quick staining for HCV, for HCV positive donors, showing a successful staining for one of the epitopes. And as you can see, I always like to mention the low numbers, which are 0.054%, and that's definitely not background. It's just this is how the populations are quite small, and that's it is what it is. But that's you shouldn't be scared of seeing such small numbers. All you have to do is just make sure you have the gatings at the right position. That's another example from which was kindly provided to us by Professor Ellie Barnes, where here in this case, because of those low numbers, they managed to culture the cells with the matching peptide, and therefore expand those cells, and the numbers have improved to 29% rather than 0.054. But in this case as well, the stainings were successful, and they were very happy with them, actually, and that's why they sent them to us. But yeah, what I need to mention as well is that all those items are guaranteed for 6 months. So if there's any issue, you can just contact us, and we'll be happy to help with the staining and give advice. Now on to something slightly different. But we're still talking about this MHC plus peptide complex relation that we are specialized in. And that's another technology that we have developed in-house, and that can help you with confirming often your in silico predictions. Now, a lot of epitopes have HLA restrictions. Certain diseases as well, like autoimmune diseases, are associated with certain HLA types. And while you can try to identify epitopes using in silico predictions, nothing is as certain as a physical assay. And this is what we have to offer. We will use recombinant MHC molecules and will investigate the binding of your chosen peptides onto the MHC molecule of choice. It's a high throughput physical HLA peptide binding assay that we have developed. We can assess the binding on 56 HLA DRDP alleles. Class 1 HLA molecules are also included. We can also look at murine specificities and simian specificities. And we can turn this around in what, 3-4 weeks maybe? That's how quick it is. So the principle is actually quite simple. We will take the MHC molecule and incubate it with peptides, and then depending on whether there is a binding or not, we will measure a signal. And that's how we can quantify how long that peptide is binding to the MHC molecule. Again, this is just a list of all the HLA types we can look at. And you can see the murine specificities for class 1 and simian specificities for class 1, murine specificities as well for class 2, if anybody's interested in those alleles. Again, to illustrate, just a quick case study using HIV, which we're very proud of as well. This was in collaboration with Imperial College. And in this case, the research group was interested in B35 positive individuals because this HLA type was associated with a rapid disease progression in HIV-infected patients. They needed to be able to monitor responses to HIV in the case of those B35 positive individuals, but they needed to identify which epitopes were binding and were restricted to B35 from an HIV protein GAG. So they used in silico predictions, came to us with 45 candidate peptides, and just asked us to load them onto B35. So that's what we did, and we reported the data. We always use a positive control, obviously, and a negative control, and we reported all the peptides that has successfully been embedded in the groove of the B35 HLA molecule. We also, of course, confirmed that all the peptides had the right anchor, the right residues in the anchor positions for this B35 HLA type, and also looked at how long this peptide was binding onto the MHC molecule. So it is one thing for the peptide to bind onto the MHC molecule. It's another thing for it to bind long enough to be a good enough epitope or a more viable epitope. So we also offer not only the binding assay as a module, but we also offer, we can also analyze the off rates of those peptide binding. So, and we test that at several time points, 0 hour, 2 hour, and 24 hours. And that also confirms how well the peptide binds and how likely of an epitope it is. So thanks to this HLA binding assay, we managed to synthesize class 1 pentamers, and that's how they managed to kind of monitor the responses to the virus by looking at the MHC class 1 responses. Just a last couple of slides, and that's a bit of a technical side of things. If you don't know the HLA types of your patients or of your cell lines, sometimes all those studies are irrelevant. So we offer an HLA typing service, and we can look at class 1 and class 2 and use several methods for that. We can HLA type tens to hundreds of samples if you need to, and the turnaround time is approximately 3 weeks or 15 working days. So that's just for your information. And again, another kind of technical part of things, and we actually specialize in the synthesis of custom peptides. We can synthesize modified peptides as well as you like in all sorts of quantities and purity. We have a high purity level available as well. And we also offer pro mixes for your ELISpot assays, if that's ELISpot assays or other applications, if that's of use. So just to finish off, as I said, the identification of epitopes is crucial not only for the development of your targeted therapies, but also for the better understanding of the immune responses generated by your therapeutics. And to understand those immune responses, immune monitoring has become an absolute requirement, whether it's through ELISpots or multimers class 1 and class 2. And at ProImmune, we can be, and we are, a trusted partner in those studies. So I'd like to thank you all and hope you'll enjoy the rest of the day and tomorrow's conferences. If you have any technical questions or would like to ask us anything about our assays, just please feel free to come to see us.